Hey everyone, Jordan Manis here with our Module 1 experiment with Gummy Bears. So I have our no salt cup here and our salt cup. Each cup has as close to 100 milliliters as I could get with the measuring cup yesterday. They've been sitting in here for about 10 hours. The salt solution, it took 1 tablespoon and 1 teaspoon of salt before it became saturated. So we have our normal size gummy bear here in the yellow, and now we're going to take out our gummy bear from the salt solution, see what happened. So we have our green gummy bear. So I'm just putting it down on a piece of paper here off the screen. I'll show you in just a second. And then we have our no salt gummy bear that actually just broke in half because it's kind of large. All right, so now we got them out of the water before they were in there too long. And so take a second and we're gonna go do some analysis.
Okay, and we're back for the analysis portion of the video. So as we saw a little while ago, with our 2 gummy bears in our water and salt water solution, the bear that was in the just water cup grew enormously in size, and the saltwater bear was a little bit smaller than what it was when we put it in, but not too off from the gummy bear that was not in either of the cups, just a normal straight out of the bag. And there'll be a picture of that at the end of the video if I can't get it overlaid here somehow. So how did this happen? So osmosis is a process that allows water in and out of cells, and in this case the gummy bear that was in the water, because it had nothing else in the solution, drew in, or through osmosis, all the water went into the gummy bear, and it grew significantly in size, as we saw. And when this happens, because there's a lower concentration or no concentration, I mean, we know there's some stuff in regular tap water, it's not 100% pure, but there's a lower concentration of dissolved substances, so that is could be said to be a hypotonic. And on the other hand, the saltwater bear is hypertonic because of the high concentration of salt that was in the solution. Again, it was a tablespoon and 1 tablespoon and 1 teaspoon of salt in that solution. So it's a pretty high concentrated solution. It's hypertonic comparatively, and it prevented a lot of the transfer of the water into the bear. So this is essentially an exercise in the differences between a hypertonic and a hypotonic solution, and the reaction it has with the gummy bear. So just in summary, the water through osmosis was collecting and was able to be drawn into the gummy bear in the water. The less concentrated solution and the higher concentrated solution with the salt water wasn't able to penetrate as much of the gummy bear and actually cause it to shrink just a little bit, not too much, as it would in a hypertonic solution. So that's all I have for this week. Thank you very much, and I'll talk to you soon next week.